Hi dear students, welcome to S&S Bioinfo. Today's topic is the Law of Independent Assortment and its explanation. The Law of Independent Assortment states that in a dihybrid cross, the segregation of one pair of characters is independent of the other pair of characters as alleles sorted into gametes independently of one another during meiosis. To learn more about the law of independent assortment, we need to consider a dihybrid cross. A dihybrid cross is a genetic cross which involves the crossing of two parents with the two pairs of contrasting traits. Now let us take an example. Here the parents are round yellow and wrinkled green. Round shape is dominant over the wrinkled shape. Yellow color is dominant over the green color. Here we have selected two pairs of contrasting traits. One is round wrinkled, the second pair is yellow green. The genotype of parent is written like this: first parent, second parent. The first parent phenotype is capital RR, capital YY, homozygous dominant pair of alleles. The second parent, homozygous recessive pair of alleles, small rr, small yy. The gametes. At the time of gamete formation, gamete receive only one allele from each pair so that the alleles are encircled like this. So, capital RY for one gamete, the next gamete also same because it is a homozygous pair of alleles. The second parent's gamete also have a same type of allele because it is a homozygous pair of alleles, recessive pair of alleles. At the time of fertilization, the male gamete fuses with the female gamete. So, the result of that genotype is in F1 plant RRYY. That means a heterozygous RR and heterozygous YY. The genotype of the plant is this one. The phenotype is round yellow. So, both dominant traits expressed in the F1 plants. So the next step is selfing of F1 plants. Selfing means self-pollination of F1 plants. So as it is a bisexual plant, the both parents have same kind of genotype. So the gametes, here you can see 4 gamete in one parent, the same gamete also seen in the second parent because the genotype of the parents are similar. So, 4 combinations can be written here. See capital R with a capital Y, capital R with a small y, small r with a capital Y, small r with a small y. So, 4 combinations of alleles can be occurred at the time of gamete formation. So, here one parent possesses 4 the different combinations of allele. That is second parent also the gametes with a 4 combination. These gametes are haploid in nature. That means they carry only single allele for a character or a trait. So to find out the possible genotype of these parents in F2 generation, so we can write those gametes in a Punnett square. That on the top of the Punnett square we must write the genotype. That means the gamete, the genotype of the first parent's gamete. So the second parent that is written over here on the left side of the Punnett square. So we need to draw 16 boxes here. 4 into 4, 16 boxes here. Then let's find out the possible genotype of the F2 plants. Here you can see that capital RY is multiplied with the capital RY. So we can write capital RR, capital YY. Similarly, we can find out the possible genotype of the F2 plants. Possible 16 genotype of the F2 plants. Here we got that 9 round yellow plants. Round 1, 2, 3, 4, 5, 6, 7, 8, 9. Then 3 round green. 1, 2, 3 round green. Then 3 wrinkled yellow. 1, 2, 3. 3 wrinkled yellow. And 1 wrinkled green. The phenotypic ratio is 9:3:3:1. Here you can see both parental type and non-parental type. The parental types are round yellow and wrinkled green. The non-parental types are round green, wrinkled yellow. New combinations of genes are occurred in the F2 generation. But there is no blending or mixing of traits or characters because Alleles assort independently at the time of gamete formation. So gamete receive only one allele from each pair. I have told you that alleles segregate independently at the time of gamete formation. Let's know more about what happens to alleles at the time of meiosis. So here I have drawn a simple diagrammatic representation of meiosis. So what is the result of meiosis? Here you can see a pair of homologous chromosomes. Homologous chromosomes are the chromosomes that we inherit from our father and mother. Here you can see blue colored paternal and duplicated chromosome. Here I have labeled A, B, D, 2 dominant genes and 1 small gene. Here unduplicated maternal chromosome. Here 3 genes, that A, small b and capital D. Here this A, B and D for 3 different traits, 3 pairs of alleles that determine the 3 traits in the progeny. See, this is a duplicated paternal chromosome. This is duplicated maternal chromosome. Duplication takes place at the time of cell division, at the time of meiosis. So duplication, so same copy of. This is a chromatid, this is a sister chromatid. After duplication, these two are known as sister chromatids. Similarly, here also duplication. So chromatid, sister chromatid, duplication of the genes. Here, after duplication, the homologous chromosome pair up and forms tetrads. So here you can see a cell with one pair of chromosomes. These chromosomes undergo, these homologous chromosomes undergo crossing over. Crossing over is the exchange of genes between the non-sister chromatids of the homologous chromosome. That means the gene can be exchanged from the chromatid of paternal chromosome to the chromatid of maternal chromosome and vice versa. And here you can see the exchange of the genes here. So that after crossing over, only I have drawn only one part that that crossing over occur only at one point. See, after crossing over, you can see the homologous chromosome pair with the different arrangement of the different set of genes. Here instead of capital A, that means instead of dominant gene, here it is a recessive gene. In maternal chromosome, instead of small a, instead of the recessive gene, it is that dominant gene. So here you can see the combinations have already changed after crossing over. So here after crossing over, that the next step is the reduction division. That means chromosome number becomes half in meiosis 1. So here one pair that chromosomes segregate to each other. So that meiosis 1. So finally after meiosis 2, one cell forms its copy as 2, again one cell forms its copy as 2. So total 4 cells are formed at the time of gamete formation. It takes place during spermatogenesis and oogenesis in sexually reproducing organism. See, each gamete carries all set of genes. See, capital A here, capital B, small d, 3 set of genes for controlling 3 traits. Another gamete that but the allele is different. Here small a, that's the recessive allele, dominant allele, again recessive for 3 traits. See another combination here, that capital A, small b and capital D, third set of genes in third gamete. Fourth gamete, then small a, small b and capital D. This is the way new combinations of alleles are formed in the gametes. So alleles sort out independently at the time of gamete formation. So gamete receive only one allele from each pair. So this gamete undergoes fertilization. That when male gamete carries one allele for a single trait, female gamete also carries a single allele for one trait. So at the time of fertilization, what happens? These alleles pair together, come together. So restoring of the alleles. So the progeny gets one copy of, that means one pair of genes for a particular trait. This is actual meaning of the law of independent assortment. Then I hope you understood the law and its explanation. Then thank you and have a nice time.